Great. Well, thank you, everyone. I'm very happy to be here speaking with you today. Cohen Veterans Bioscience is, if you're not already familiar with us, it's a nonprofit research biotech, and we're dedicated to fast-tracking diagnostics and therapeutics for all those suffering from trauma-related and other brain disorders. I don't think I really have to make the case for furthering research on brain health, but I don't know how many of the people in attendance work in CNS. There are over 1,000 diagnosable CNS disorders that directly impacts about 4 billion people and has a tremendous societal cost. Unlike some of the other disorders we've talked about today, there really is a lack of objective diagnostics in many of these disorders, especially when you think about psychiatry. And that is a tremendous contributor to the lack of precision therapeutics in CNS. So what I'll talk about today is really how CVB is trying to move psychiatry closer to precision medicine. I won't spend a lot of time talking about the need for a new approach. I think that's pretty obvious. I'll spend most of my time talking about all the different biotyping efforts that we have going on in partnership with many different organizations. And then because digital data and wearables and biosensors seem to be very popular and moving into all kinds of clinical trials, I'll spend a little bit of time at the end talking about those and some of the challenges and some of the things you have to consider before you include them in your clinical trials. So at CVB, we've started, we're interested in all brain health, but we started with a focus on PTSD. It has a high lifetime prevalence of about 8% in the general population, and we know there are certain populations where this prevalence is even higher, the military, for example. Psychotherapy is the first-line treatment, but it is underprescribed. When it is prescribed, it's underutilized, and for those who start it, many don't complete it. In terms of drugs that have been approved, there are only 2, Paxil and Zoloft, and for a disorder that has such a heterogeneous population, these are both one mechanism. They're SSRIs, so targeting only one biological pathway in that heterogeneous population. And surprisingly, those were approved over 17 years ago. So clearly, there's a failure somewhere along the line. I'm sure many of you have seen this slide before. This is from 2015, and it's taking a look across all diseases. This is not just CNS, and all modalities, and looking at the likelihood of success for any therapeutic that gets into clinical trials and the likelihood of getting to approval. And it's fairly low. It's 1 in 10. When you take a look at psychiatry, it's even lower. It's about 6%. And I mentioned the heterogeneous population. In psychiatry, so I'm sure some of you know, the diagnostics are often clinical interviews asking about symptoms, so they're subjective. In PTSD, it's the same, and it's across multiple different symptom domains, and patients can have a combination of symptoms from each of these different domains. And the gold standard for the outcome measure in PTSD is this exact same scale, so it's not the ideal situation. And Isaac Alter-Levy did a study looking at all the different possible combinations of these symptoms you could have and still reach a diagnosis of PTSD. With over 600,000 different combinations, it's not a surprise that when you're testing a single biological mechanism in a treatment trial, and maybe 50% of your patients don't even have that underlying biological mechanism, there's going to be a ton of noise and you won't be able to move your products forward. So we need a different approach. CVB started at the beginning doing our homework and saying, okay, there's a ton of research in this area. We did a landscape to see how many different biomarkers there are identified for PTSD. We were excited to find that there were over 750 biomarkers in PTSD. And this is thinking about diagnostic biomarkers, biomarkers predictive of treatment response, or prognostic biomarkers, and across multiple modalities, imaging, EEG, genetics, et cetera. And so I just have for a little bit of audience participation, poll question here. Of these 750 PTSD biomarkers in the literature, how many do you think have been validated? Sam, I don't know if you're able to put up the response question for that.
So we have it up on the screen, ladies and gentlemen. How many PTSD biomarkers, which includes fluid imaging, EEG, physiopsychological, genetic, epigenetic, have been validated to date? She's given everybody a couple options: 0, 2, 6, 11, and 25. So, Dr. Gage, I'll give everyone another 10 more seconds. Okay, 7, 5, 3, 2, 1. Okay, and looks like majority have chosen 2.
Okay, well, maybe it's not a surprise. 2 is pretty close. Oops, do I have this back? It's 0. None have been validated. And so it's clearly time for a different approach. It was nice to hear some of the presentations earlier about the benefits of complex, innovative designs versus traditional randomized controlled trials. And to do those, you really need to pair those with objective endpoints rather than the subjective endpoints we typically have in psychiatry, and perhaps starting to look at constructs rather than the single diagnostic population we've been thinking about. And certainly to get to precision medicine, we need to have deep phenotyping of these patients going in order to start discovering some of these biomarkers rather than relying on serendipity as we've done often in CNS. And so at CVB, we try to embody this paradigm shift. And our approach is to build enabling platforms with strategic partners so that we can adopt a team science approach and move these things forward in years rather than the decades that it's often taking us. We've built. We started at the beginning with data standards. It may sound a little bit like a boring topic, but it actually was very exciting to partner with CDISC and build the first PTSD data standards, not just for pharma, in case they happen to enter into the world of PTSD and test a therapeutic, but we also try to bring this to many of our academic partners so that the data can be combined across their different studies. We have built a cloud-based data commons with the capacity to store, secure, and analyze data so that we can foster that team science discovery approach. There has been a lot of research in PTSD. It's not like it hasn't occurred, and there are many data sets out there that we're trying to bring together. And you need something like this Brain Commons to start combining these data across studies. And it's a nice resource. Each partner has their own space where they can analyze data right there on the cloud. When we're talking about genomics or imaging data, we're talking about petabytes of data, and you really can't feasibly download that and analyze that on your own systems. So we've built a place where you can analyze it there. And then we think about trying to bridge the translational gap. You often think about animal models, and in psychiatry there are many models, but none of them are validated models of psychiatric disorders. And so we've started partnering with different academic institutions and different CROs to validate some of the models that are most commonly used, and then to industrialize them. So when you're testing your therapeutics in these models, you know exactly what your results mean and how they might predict behavior in a clinical trial. We also did some work on the infrastructure. There are many small trials out there, and everybody's analyzing things on their own assays. And so we started, one of the nice things about the legacy data sets that I mentioned is that because of their method of funding, often they have biosamples still available that they never had the money to analyze. And we've been able to partner with them and bring them into our biorepository and biobank so that they can be analyzed at the same time and on the same platforms to try to reduce some of that pre-analytic variability. And then the last one I'll mention is our RapidDX platform, which is a platform to discover and validate biomarkers. And when I'm talking about biomarkers, I'm not just talking about genetics or fluid biomarkers, but we really have the technology now to quantify brain health and to do so in a way that is not just getting a blood draw at a clinic in a clinical trial once a month, but really collecting continuous data from patients in their real lives and doing so in a passive manner. And so taking a systems approach to collecting all of these different potential biomarkers. And so what I'm showing here is really just a schematic of the approach I've been talking about, taking all of these multiple modalities to a very heterogeneous population and start using them to separate the groups out into these different clusters, and then continuing that and replicating that in another population, which you can then take a look at the biological mechanisms and match those actually to drugs that target those mechanisms. And you might notice that the title of the slide is to move away from the DSM diagnosis. And that's a little bit far-reaching and grand, but it is something you need to consider when thinking about biomarkers. It's not just discovering and validating them, but what is their likelihood of approvability? Can they be used as an outcome measure? Will they match up to what the current gold standard is? And so you have to think about context of use and the regulatory approach that you'll use going forward. And so does this work? The same study I showed you before that showed that 1 in 10 products makes it to approval. They also looked at studies that had actually used biomarkers to select the population. And what's nice with just looking at biomarkers, you'll go from 1 in 10, you get a 3-fold increase in the likelihood of success. So just with biomarkers alone, you can improve your success level. And so the last platform I'll mention is our VIA platform, standing for Validator, Incubator, and Accelerator. And it takes all of these approaches together in partnership with different pharma companies or academics, anybody that wants to allow us to help them use a precision medicine approach. And we can reduce the risk early on so that we can increase the likelihood of probability going forward for each of our partners. So I'll focus on our RapidDX platform now and just show you very briefly 4 different programs that we started and have ongoing. The first one that we did really took, this is the roadmap for RapidDX that you're looking at. And it really takes you through just building that infrastructure out, making sure that we think about our harmonizing methods at the start, thinking about what context of use would be feasible once you get to the clinic, before thinking about what is the design of the study you want to do, and then replicating and bringing it to the clinic. And the first program I'll show you really took advantage of some of these infrastructure pieces of methods harmonization, establishing public-private partnerships, and really thinking from the beginning, what is the statistical plan you need before you start bringing in data. And so for us, we really think that genetics is at the start of discovering the biological underpinnings of PTSD. And there was already the Global PTSD Genetics Consortium established, and they had collected a number of cases and controls from a few sites across the country, and were slowly trying to build up their numbers. And they had their first freeze that didn't have any genome-wide significant hits. We talked with them and other partners such as the Stanley Broad Psychiatric Research Center, and said, well, let's think first about how many cases you actually need in order to get to a likely place where you'll get to an inflection point where you start seeing genome-wide significant hits before we just start collecting samples. And we did that analysis looking at other disorders like depression and psychiatry, and then purposely went out and worked with over 130 collaborators across 12 different countries and brought in over 70,000 samples. With that data, we recently published an article showing that we did find 6 genome-wide significant hits, and estimate the SNP-based heritability of PTSD to be about 5 to 20%, which is correlates well with depression and other psychiatric disorders, showing a susceptibility risk for PTSD. The next program I'll talk about was really thinking about fluid biomarkers. And in that biomarker landscape that I talked about, a lot of the research focused around inflammatory biomarkers and cytokines. And while that's an area of investigation that many researchers follow, the data out there was conflicting in every way possible, and things just didn't replicate. And so we started to look at the reasons for that, and we focused on the assays that were being used in the studies. We talked with 17 different vendors about their assays for inflammatory biomarkers, trying to understand what some of the challenges are and how the different assays may contribute to the conflicting data. And the inflammatory markers are at very low concentrations, and so the analytical sensitivity you need is extremely high. Among the 17 vendors we talked with, there were 5 that really seemed to meet the clinical and technical aspects needed. And we worked with those 5 vendors to conduct a bake-off. We collected 72 clinical samples from PTSD patients, Parkinson's patients, and healthy controls. We spiked those with some known standard samples and then sent them off in a blinded manner to the 5 different participating assay vendors. Each of those vendors ran samples twice and then sent the data back to us for analysis. I'd say it's surprising, but when you look at the studies in the literature, maybe it's not so surprising that there was huge variability across the different assay platforms. And we think this is really key to understanding how you can discover biomarkers. I'm just showing you here results looking at the precision, the coefficient of variation for the different assay platforms, as well as the sensitivity of those. And you can see how highly variable they are. So that was for inflammatory markers. We currently have a bake-off, similar bake-off going on for vendors who have assays in metabolomics. And those results will be available at the end of this year. And then we have bake-offs planned for other assay platforms in transcriptomics, epigenetics, and exosomes. The last program I'll show you was looking at trying to identify a neural signature on FMRI of potential treatment response to psychotherapy. We worked with an investigator at Stanford, Dr. Amit Etkin, to study biomarkers on FMRI And he worked with a population of PTSD patients that when you looked at their symptoms, you couldn't separate them into any particular groups, but yet they had very different responses to psychotherapy. He had already had several different small studies that had led him to believe that he had a neural signature, a biomarker on FMRI of those who were not likely to respond to psychotherapy. And we worked with him to replicate that data in a larger data set. And this was published this past year, or at the end of last year. And this is just showing you the large effect that he saw replicated when looking on FMRI at the ventral attention network. The patients separated into 4 different groups, only one of which did not respond well to a type of psychotherapy called prolonged exposure. So you see the blue group. They are those that had low connectivity in that network, and also on a verbal test of memory were impaired. When those 2 things were combined, not one alone, it separated that group in a replicable way into those that did not respond to psychotherapy. And so this, you can imagine, would be a huge benefit when somebody's coming in, and rather than put them through 3 months of prolonged exposure, which is a type of therapy that can be effective, but people do not want to go through. Typically, people with PTSD do not want to think about the trauma that they've experienced, let alone really discuss it in a way that brings up all of these memories over such a long period. And so if somebody's unlikely to respond to this, then you can start thinking about different ways to work with them. Dr. Ekin actually spun this out into a company called Alto Neurosciences, and we worked with him to make this more scalable. FMRI is not really something that's scalable to such a large population, and he has now found the same signature, a signature on EEG that represents the signature that was on FMRI, so a much more scalable approach. So digital biomarkers. We've been working with an organization called MyHelios to improve a common therapy for insomnia. Insomnia is a common symptom within PTSD. And so the therapy that's very efficient is cognitive behavioral therapy for insomnia. And when patients follow all of the instructions and are able to accurately report their sleep patterns back to the clinician, it can be very successful. And so CVB's approach is to say that the gaps of not knowing whether the patient is actually following the instructions. For example, you're only supposed to stay in bed for a certain amount of time if you're not falling asleep. So you're supposed to get back out of bed if you've been in bed for more than 20 minutes and haven't fallen asleep. The clinician doesn't really know if the patient is following that instruction, and then they're providing sleep diaries talking about their sleep patterns, and patients are not very accurate in being able to record that information. So we're instrumenting CBTI with different wearable sensors and other biosensors, so that that data can be analyzed and presented in a way that the clinician, on a dashboard, the clinician can use that information to then guide their subsequent treatment sessions with the patient. But you can't just throw any digital solutions into something like this. You really have to understand the devices. You have to think about what is the information you actually want to collect. How are those data collected? Are they directly available to you? Are you getting raw data? Do you have to create an app to actually see either the derived variables that come from those devices or the raw data? And how do those match up with the outcomes you'd like to think about? And you have to really think about how accurate these devices are. And so at CVB, we have a device lab that looks at all of these things I just mentioned, thinking about the data pipelines for these devices, determining whether they're precise. So precise, thinking about how consistently you'll get the same answer for the same action. So for example, if you walk 10 steps, does it say 10 steps every single time you do that? Whether 10 steps, maybe it says 9 steps every time you walk 10 steps, but it does that every single time. That's precision. Accuracy is thinking more about sensitivity and specificity of the device. And we, in the device lab, we have already evaluated potential devices for precision, and we're now conducting a study that looks at the accuracy of the devices that met all those other conditions in a technical verification study with the University of Pennsylvania. But the accuracy and precision of the device is not the only thing you have to consider. There are many other things you have to consider when bringing a biosensor into your clinical trials. The first is, are you going to get any data in the first place? If it's not comfortable and the patient doesn't want to wear it, you're not going to get any data. If the battery life doesn't last all night while they're sleeping, you're going to lose data. What is the actual storage capacity of the device if you're only going to be downloading the data maybe once every month when they come into the clinic? How are you going to think about, let's say, the safety information that might be transferred? You need to make a decision. Are you going to look at this in real time and potentially think about it as a safety signal? Or are you going to make sure that the clinician and the patient understand that these aren't going to be used for safety evaluations and they shouldn't think about it in that manner? And then there's also always bias. Is it a device, let's say it's a Fitbit that you're using, is the patient able to see the data? And are they going to change their behavior because they see that data? And is it just that feedback loop that's making them seem to improve versus the therapeutic you're trying to test? So I mentioned that in our Sleep Well program, we did put many devices through the paces in our device lab. And we came up with 6 different devices that we're going to be testing in our technical performance verification study. 3 different wrist wearables, a ring, and 2 different sleep mats. And we're comparing these individually against the gold standard, which is polysomnography. And so this study has just started at the University of Pennsylvania. Subjects come in and they wear all these devices and sleep on the sleep mats overnight, and they do that twice. And then all of the data come in-house to CVB, and we analyze those and try to develop algorithms that might even put some of these devices together, let's say a wrist wearable with one of the sleep mats, to achieve the accuracy of polysomnography. And if so, then you can move on to a proof of concept study with CBTI and actually test whether you can now get this information consistently over time from the patient at home, which would be a great step forward. So the last thing I'll mention, which is fairly new and is very exciting to me, is really using artificial intelligence to identify digital movement-related biomarkers. So we started with a proof of principle study in a condition where we thought the motor movements would be more easily picked up, just to start off with something that we thought would be easier. So we worked with, we thought about RETs, where the girls that have this condition develop hand stereotypies, And there are about 22 different hand stereotypies that they show. This slide just shows 6 examples of them. And we selected a wrist wearable to pick up those hand movements, the Shimmer 3 IMU. And we started with healthy volunteers who learned 4 classes of these hand gestures. So the stereotypies that I just talked about, but also normal behaviors such as brushing teeth or moving the fork up and down, simulating writing, also no movements, and then some aggressive hand gestures. These were recorded, and then the data were analyzed to see whether we could develop a deep neural network to discriminate between each of these gestures. And this was successful. It was recently presented. The training accuracy of that algorithm was about 92%, and then a subsequent validation set had an accuracy of 84%. So that's the start. And then we've paired up with the Children's Hospital of Pennsylvania to move from that proof of principle study to a proof of concept. Is it actually feasible to collect this data from girls with Rett's? And in real life situations, can you distinguish these movements, not just pick up movements that are purposely being done by a volunteer? And so we're videotaping Rett girls that will automatically demonstrate some of the stereotypies, but we'll also have sex and age match controls be videotaped, making their typical, either not moving or their normal gestures, demonstrating some aggressive gestures. So for example, some of the stereotypies might be some very fast clapping from the Rett girls, which of course, at some point, the normal girls will be doing the same thing, but the frequency, the speed, that is likely going to be different. And so these recordings will be used to add to the general model that we built from the proof of principle study, but it will also be used to develop a personal model for each of the individual Rett girls. And that's because each girl, typically, they won't do all 22 hand gestures, they'll only do a few of them. And so you need to develop a model for each one of them. And while we're mainly assessing feasibility in this study, we're also exploring the ability, if we are able to collect the data and build these algorithms, to see if we can use this as an objective assessment in a treatment, as a treatment trial outcome measure. And then if that's successful, you can imagine there are many other disorders that this could be extrapolated to. For me, when I think about PTSD, a common symptom is nightmares, and we have no idea yet how to really assess nightmares. But perhaps this can pick up different movements, distinguish between restless leg syndrome, that's another common symptom, comorbidity in PTSD from nightmare movements, for example. So at the end here, and I think all of these have to come together in the end into clinical trials. And our approach really is to think about some of these complex innovative designs. And in order to do that, you really have to do all of that deep phenotyping of your patient population at the beginning. And we think the most efficient approach to move forward is using an adaptive platform trial. There are many therapies out there that have been suggested for multiple different biological mechanisms suggested for PTSD, and we think it's the most efficient way to go forward. I won't go through the different advantages of an adaptive platform trial because it was spoken about twice here, but we think it is the best way to move forward, both the adaptive part of clinical trials where you learn from the data you collect as you go along, but then also to continually bring on new medications as you make the decision to drop drugs or move them forward into a next phase trial. That's it. Thank you.
Wonderful, Dr. Gage. That was a very insightful presentation, and definitely what you and your organization are doing for veterans that are suffering from PTSD and other, CNS related illnesses is quite remarkable. So if there's any questions from the audience, and we do have one from one of our own, Dr. Jeffrey, right here. Which of the biomarkers you described do you think shows the greatest probability of being an approvable endpoint?
So I think that I think the inflammatory markers, while they show as a very common biomarker to pick up, I think it's going to be something that's going to be across disorders and common. I don't think that'll end up being something that's approvable on its own. Likely we'll end up with several different biomarkers together. But of the ones I've shown, I think it's going to be the EEG neural marker. And the study that was done, I think that it has difficulty when you think of the regulatory perspective. It's a huge effect, but when you consider the context of use, I think you have some difficulty, because what do you do from when you go to the clinic and you have someone who is suffering from PTSD and first-line therapy is psychotherapy, and you find that they're very unlikely to respond to psychotherapy. That's a hard context of use to have. You might think about it in the opposite direction. Here's the 75% of people who are likely to respond to psychotherapy, and you can encourage them to stick with the psychotherapy when they might not want to because of that. But what we're hoping, and we're embarking on some other efforts to take that same neural signature, maybe it's a signal of response, and maybe we can use that also with different types of therapeutics.
Wonderful, wonderful. So yeah, I believe Dr. Gage's slide deck will be available, if I'm not mistaken. I believe you have already shared it with myself and Sherry. So if you're interested in learning more about her organization, you can visit them on the web. Cohen, thank you very much for the presentation.
Thank you.